Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork perspective. In this video we will talk about the DNA structure. That's very easy and quite simple. So the DNA structure I will first of all concept about the here is the cell, plant cell, which that are contained nucleus and vacuole and organelle, and here is the animal cell which that contain nucleus, organelle and little molecules in the color in the shown the bubble like structure. So nucleus contain DNA material which that are in the form of genome. That is the genome is condensation form is known as chromosome. That's here is the mitochondria which that are another organelle which that are contain another their own DNA in the form of ring that are shown in the red color, and another organelle which that are chloroplast which that are producing energy, they are producing sugar, and that's contain also ring. That's the chloroplast which that are present in the plant cell but not in the present in the animal cell. So here's the thylakoid membrane which that are very important function. There's the absorb the light in the presence of chlorophyll. That's chlorophyll are present in the thylakoid membrane, and there's a green pigment shows that during the presence of magnesium, and that's absorb the light and produce energy. There's the light energy produced as the chemical energy in the form of sugar. So now we will talk about the here is the nucleus which that contain nucleolus and chromosome. There's a 46 chromosome in the pair form are present in the human being, and there's the 46 and 23 pairs of the chromosome. So here's the nuclear pore which that are used for the messenger RNA going and transfer RNA, as well as the ribosome going from the outward into the cytoplasm for translation. So here is the chromosome which that are contain centromere, sister chromatids. That is the basic structure of the chromosome. So here's the chromosome will cut from here, and there's the uh, heads of chromosome made up of supercoiling of the uh, genetic material that is in the form of supercoiling, and we will further check their thread is shown here. That is the coiling between within the supercoiling. That's the coiling within the supercoiling is known as basically that's supercoiling. That is the DNA are present in the form of coiling. So here's the chromatid that's shown in the figure, and that we will take a one thread of the um, here is the genetic material with that one thread. The one thread is the known as the chromatid fiber. Yes, that fiber is the fiber like structure which that are further classified from here. So now we will talk about the here is the basically histone protein which that are bound by a linker protein to sustain the basically maintain the structure of the DNA in the condensation form. So here's the basically histone protein which that are showing in the figure, which that are condensedly interact to each other by a wrapping around the DNA and linker protein which that are maintain the genetic material in the histone protein which that are linker protein which that are linked together. And there is the histone protein 4 type: 1, 2 and 3 and 4. That's the 4 type of histone protein which that are bound together as a tetrad. So here we will draw a magnification of the DNA that's in the form of here. That's the helical structure of the DNA which that are helical due to the hydrogen bonding. So here the DNA which that is contain a backbone of the DNA double stranded DNA mean deoxyribonucleic acid. So first of all we will understand about the deoxyribonucleic acid mean the meaning of the DNA. So now that's we can get the concept from the biochemistry. So the biochemistry of DNA is the basically ribose sugar, that is the pentose sugar, because pentose mean the 5 carbon are present in the single pentose, that's in single ribose. So here's the deoxyribose nucleic acid mean the deoxyribose are present in the DNA. The deoxyribose mean the carbon number 2 which that are contained without oxygen. And here's the ribose RNA that's present in the RNA which that contain carbon number 2 is the basically hydroxyl group, and deoxy means the not hydroxyl group are present in the carbon number 2. So another component is known as the phosphate, that is the phosphate group that is contain hydroxyl group, and 3 oxygen with that are 1 double bond and can be double bond, and that is the basic structure of the phosphate. So now we will talk about the here is the nitrogen species which that are also present via using for the hydrogen bonding. That's a using of the hydrogen bonding by nitrogen species, that is the adenine and thymine. That's the double ring structure is known as purine. That's purine means the 2 nitrogen bases adenine and guanine that is in the form of 9 head edges, the 9 number of the basically here is a carbon and nitrogen containing 9 component which that is the here, and that is known as purine. That's a double ring, and nitrogen are present is upper side and outside, we will discuss in later. So here is the basically no and single ring we will talk about the here is the single ring cytosine and uracil and thymine. The cytosine is the basically the structure is 6 carbon and nitrogen edges, and here is the RNA only, uracil, and while the thymine DNA only. So that RNA only uracil are present which that are in the form of oxygen, and there's the bond is present is here, and here is the basically ribose which that are RNA only, and here is the adenine and guanine, there's a purine, and here is the single ring which that are uracil, thymine, and cytosine is known as pyrimidine. That is here is the pyrimidine because single ring, and here is the double ring is known as purine. Now here we will talk about the structure with the backbone of the and nitrogen bases attachment is here, that is the basically ribose sugar with that are deoxyribose sugar are present is phosphate group with that are present in the form of phosphodiester bond. That's phosphodiester bond mean oxygen in upper side and oxygen in downside between the phosphate group is known as phosphodiester bond, that is known as phosphodiester bond. And here is the D-ribose pentose sugar that is present is basically which that are attached with the carbon number 5, and there's another end is the carbon number 3, and here is the basically deoxyribose nucleus is here because hydroxyl group is not present. So remember it, that's hydroxyl group is not present in the carbon in the DNA, that is why it's known as deoxyribose nucleic acid. So here is the backbone of the phosphate group which attached with the sugar, and that sugar carbon number 1 of the sugar with attached with the nitrogen base is thymine is single ring and adenine is double ring. So there is the hydrogen bonding between hydrogen and oxygen, and hydrogen with the nitrogen is the hydrogen bonding is present. So now we will learn about the hydrogen bonding is here. Thus hydrogen bonding is a very weak bonding. It is the basically it is the weak bond which partial intermolecular bonding or interaction between one part of an electron rich donor atom. It means nitrogen in a rich electron, oxygen is rich electron, and fluorine is rich electron is the major example of the hydrogen bonding with a hydrogen. So here the water we will talk about is containing with the oxygen, and oxygen is a too much electron which that is in the form of pair, and here the basically partial positive charge in the hydrogen and partial negative charge in the oxygen that's attract to each other, and there's not completely donate the electron. So semi-donation of electron, for example, there's a relation between that donation that is known as hydrogen bonding. So here we will talk about the carbon number 1 2, the attachment is here. So thymine and adenine will bond two hydrogen bond only, while carbon and basically cytosine and guanine will binding with a hydrogen bonding, which is three hydrogen bonding is present is here. That is a adenine, and here is the thymine, and here is the bonding is three. So three mean nitrogen and oxygen and hydrogen. That is the basically three carbon to guanine, cytosine and guanine, sorry. So here is the basic structure of the we will draw here is the whole structure of the bonding. So guanine cytosine will form guanine cytosine 3 bonding, while adenine and thymine will 2 bonding, I'm sure. So here is the adenine thymine 2 bonding and guanine and cytosine 3 bonding. But in the case of uracil, they are present in the RNA. No, we will discuss about the DNA, but not RNA. RNA is the I will give the video in the description box about the RNA. Then you can watch it. So here is the basically adenine and thymine in every time double bond of hydrogen bonding, and guanine and cytosine is 3 bonding usually. So here is the 3 bonding, here is the 2 bonding with the adenine and thymine, and here is the basic structure of the later we will discuss about it. So yeah, here is basically 2 bonding, here is the 3 because guanine and cytosine, GC AT GC, and you can remember it, and here is the GC, and here is the AT or TA AT basically TAAT. You can remember this like at and ta, and you can remember about that the GC or CG. So here the backbone is contain phosphate group which that are also sugar attaching. So here now we will discuss about the phosphate group which that are phosphodiester bond are present is here. That is the here due to the two oxygen containing phosphorus. Between the phosphorus and oxygen is upper side and lower side, that is known as phosphodiester bond. That phosphodiester bond is very tight as compared to the hydrogen bonding. So that's why the DNA can be easily converted into the RNA by providing heat or something which that are used in the polymerase chain reaction which that are giving the temperature. And here is the basically the structure of the backbone of the phosphodiester bond, which that are carbon and the number 3 to attach the carbon number 5. So that's mean the carbon number 3 to 5 is the toward the 5 carbon, and here is the 3 prime to 5 prime conversion. There's a moving, there's the anti-parallel. Both strands are anti-parallel to each other. It means the basics. I will give you the concept about the anti-parallel of the structure of the DNA. That's the anti-parallel mean because here's the carbon and number 3 attachment of phosphate group, and carbon number 3 attachment of phosphate group is the basically toward the 5 carbon number. There's the phosphate group, and there's the reverse as compared to the anti-parallel in the other hand. So the purine which that are double ring nitrogenous base. So I remember I will give you a memorization, and that's always bind with single pyrimidine. It means the double ring is the basically combined with the single ring nitrogenous base. It means the nitrogenous base is the basically double ring also attached with the single ring by a hydrogen bonding. So here, so there's the important things which that are showing in the figure. So here we will talk about the helical structure, but due to the hydrogen bonding by the attraction to give a helical structure. That's helical structure is due to the double bonding and triple bonding in the DNA. That's a double bonding and triple bonding is give the significance attraction. For example, triple bonding more attraction and single bonding light attraction as compared to triple bonding. So that's the helical structure will be formed. So thanks for watching.